Hello friends, today I'll be talking about genetic code and how the genetic code was deciphered and what are the properties of genetic code. Okay, so first let me define. So what is the genetic code? The genetic code is the relationship between the nucleotide sequence of the DNA or the nucleotide sequence of mRNA with the amino acid sequence of polypeptide or protein. Okay, so now we need to first understand what is a gene and what is its information and how this information is carried to mRNA. Okay, so this is a DNA. So a particular segment of DNA is called a gene. And what is its information? That the precise nucleotide sequence in the DNA is its information, and this information is carried by a special type of RNA that is called messenger RNA. Okay, so the information present in this mRNA is converted to the sequence of amino acids, the sequence of amino acids in protein. Okay, we know that the proteins composed of several hundred of amino acids, several hundreds of 20 different amino acids. A protein is composed of several hundreds of 20 different amino acids. But in mRNA, we have only 4 types of nitrogenous bases, that is U, C, A, G. In mRNA, we find these 4 types of nucleotides. So, and in proteins, we have 20 different types of amino acids. So, how these 4 types of nucleotides codes for 20 different amino acids is the main basics of genetic code. Okay. So, to understand this, we first go hypothetically how the code was deciphered. So we'll take some cases. So case 1, first we'll take case 1. We have only 4 types of nitrogenous bases or nucleotides, that is U, C, A, G. Okay, and these are the hypothetical views. Okay, so if one nitrogenous base codes for one amino acid, then these 4 types of nitrogenous bases would cover only 4 amino acids. These 4 types of nitrogenous bases will only code for 4 amino acids, which is insufficient because in proteins there are 20 different types of amino acids. So the singlet codon or the singlet code is not possible, right? Okay, now we'll take case 2. Here, the sequence of 2 bases, we will combine 2 bases. The sequence of 2 bases like AU or UC or GC in this manner. We will take in this manner the sequence of 2 bases. They will form a 16 types of combinations. 16 types of combination. Okay, 16 type of combinations. So because we have 20 different amino acids. So this is also insufficient. So the doublet code is also impossible. Okay, and then now we'll take case 3. The sequence of 3 bases. Here we'll combine 3 bases. So they form a total of a permutation combination, that is it brings a total of 64 combination. When we take 3 bases, when combine 3 bases, so they will form 64 combination. Okay, this 64 combination codes for 20 different types of amino acid, which is sufficient. So the genetic code consists of 3 nucleotides. 3 nucleotides means a codon consists of 3 nucleotides. 3 nucleotides specify a amino acid, and these 3 nucleotides is called a codon. So it was George Gamow who first stated that the code consists of three nucleotides, means a codon consists of three nucleotides, right? So thereafter, many of the molecular biologists like Nirenberg, and another Hargobind Khorana, and Mathai, they supported George Gamow that a codon consists of three nucleotides. They experimentally proved that a codon consists of three nucleotides. So let's discussion have a discussion on Nirenberg's work. So how Nirenberg deciphered the genetic code. Okay. Nirenberg constructed a poly-U-mRNA. Poly-U-mRNA means this mRNA consists of only uracil nucleotides. Okay. And allowed for translation. He found that the protein, the amino acid found, that is the phenylalanine, means the UUU, this triple U codon, codon codes for the amino acid phenylalanine. Suppose this mRNA composed of 13 nucleotides. If this mRNA has 13 nucleotides, so the number of amino acids found in this polypeptide was 10. Means 3 nucleotide codes for specified a single amino acids. And this UUU, this codon codes for specified the amino acid phenylalanine. So now we'll go to the Khorana's work, Hargobind, Dr. H.G. Khorana, an Indian-born American scientist. So Khorana constructed an mRNA that consists of CUC, UCU, CUC, UCU. So non-nucleotide sequence. He constructed an mRNA containing such type of nucleotides. So CUC, UCU, CUC, UCU, in this manner. Okay. And he allowed for translation, and it found that in protein, the amino acids found were leucine, serine, then leucine, serine, in this manner. So the CUC, this codon codes for leucine, and UCU codes for the amino acid serine. Okay. In this way, they decipher the genetic code. Okay, now we'll discuss about the properties of genetic code. Properties of genetic code. The genetic code has certain well-established properties. So let's have a discussion. So number one, the code is triplet, means a triplet code. The code is triplet, means each codon consists of three nucleotides. Triplet means each codon consists of three nucleotides. In mRNA, three nucleotide means a nucleotide triplet. Nucleotide, it is also called nucleotide triplet, is called a codon, means each codon consists of three nucleotides. Okay, the second property, the code is degenerate. So we have 64 codons. If we exclude 3 termination codon, then we have termination. If we exclude 3 termination codon, then we have 61 codons. And these 61 codons codes for only 20 amino acids. Okay. So one amino acid is specified by more than one codon, because the number of amino acid is 20, but the number of codon is 61. So one amino acid is coded by more codons. So this property is said to be, this genetic code in this sense is called degenerate. I am citing an example, some amino acids that are coded by some amino acids like serine, arginine, leucine. They are coded by, specified by 6 codons. They are specified by 6 codons. And third property is non-overlapping. Non-overlapping means no single base can take part in the formation of next codon in a succession. Okay, so I am citing the example here. Suppose CCU GCC. It is read as CCU and GCC. So it is non-overlapping. Okay, and the overlapping codons have been found in 5 cross 174 coliphase. It is virus. Okay, here I explain what is overlapping, and what is non-overlapping. Okay, right? Suppose it is CCG and GUC. So CCG, then if the next codon starts from CGG, and then the next codon is GGU. So in this manner it is overlapping codon. But if we take CCG, GUC, and so CCG, then after CCG comes GUC, here no single base overlap. So that is non-overlapping. So the genetic code is non-overlapping. Then the fourth property is non-ambiguous. Non-ambiguous means each codon has only one meaning. Each codon has only one meaning. Means each codon has specific sense for its amino acid. It never codes for any other amino acids. Okay, suppose one example, that is suppose triple U, it codes for phenylalanine, and it never works for any other amino acids like serine, arginine, etc. Okay, so one exception of this non-ambiguous codon is GUG. So this codon, this GUG, it normally codes for valine. It codes for valine, but at the initiating position, at the starting position, means when AUG is absent, the initiating codon, the absent GUG acts as the initiation codon. At the initiating position, it codes for methionine. So because of this reason, this GUG codon is ambiguous. Ambiguous codon. Okay. So number 5, the code is commaless. Means the codon is read in mRNA in a contiguous fashion. All the codons are read in mRNA in a contiguous fashion. There is no comma or punctuation between successive codons. Then the sixth property, that is the starting codon. The starting codon is AUG. So it is called the initiation codon because it starts the translation process. In mRNA, the first codon is found as AUG. It has dual function, so it acts as initiation codon and it also codes for the amino acid methionine. Acts as initiation codon and codes for the amino acid methionine. In case AUG is absent at the initiating position, then GUG acts as the starting codon, and GUG also codes for methionine at the starting position. Okay, this is the starting codon. Okay, then the seventh property, that is termination codon. Termination codon. So this codon terminate the process of translation. This codon terminate the process of translation. They terminate translation process. Okay, so there are three terminations codon: UAA, UAG, and UGA. It is they are called ochre, amber, opal. So they give signal the end of the translation process. This give signal the ending of the translation process. These three codons are called termination codon or stop codon or nonsense codon. Okay, so number 8 properties, that is the code is universal. Universal. Okay, this genetic code is applicable in all the living system. Applicable in all living system, whether it is plant or animals or microorganisms Since this genetic code is applicable to all the living organism, it is called the universal genetic code. Okay? Okay. I hope this video will help you understand the genetic code. Thanks.